HP1 alpha is an interesting protein. It has a chromodomain that binds to the methylated lysine 9 of histone H3. This is how it localizes to heterochromatin. It has a hinge region that's known to interact with both RNA and DNA, and then it has a chromoshadow domain that's important for its protein-protein interactions. And so we'd like to know, does CBP alpha interact with HP1 alpha? This is a imaging experiment where we have a YFP labeled HP1 alpha, a CFP labeled CEBP alpha, and this is of course the merged image showing that they exactly co-localize with one another. This is a pull-down experiment, a co-IP experiment, where we've pulled down CEBP alpha but then blotted for the epitope tagged HP1 alpha, and you can see the two come down as part of a common complex. So it appears that they do indeed interact in some form of a complex. But what we'd like to know is how are they interacting, and using live cell imaging approaches to try to get at the specifics of those interactions. So as I mentioned at the beginning, we're using FRET approaches to look at the steady state interactions of these proteins, and we're using fluorescence correlation spectroscopy approaches to get at the dynamics of these interactions, whether these proteins interact as part of a mobile complex. And so I'm going to first address the FCS approach. And the system we have is a ISS ALBA system that's set up for doing FCS. It's on an IX71 microscope. We use a 60x 1.2 NA water immersion objective for our imaging. We have a pathology device stage top environmental chamber that controls the temperature at 37 degrees. And we have two different laser lines that we're going to use for FCS. We have a 448 laser line to excite cyan fluorescent proteins. We have a 561 line to excite red fluorescent proteins.